Whose microbiome is it anyway? Obesity Explained, Episode 5, featuring the bacteroidetes to firmicutes ratio. In the last episode, episode 4, I presented the idea that fattening is a biological process, and we looked here at Otis, the Fat Bear Week champ. We see that Otis needs to fatten up for winter to survive, and so fattening is the biological imperative of the hibernator. This chart shows mammals who actually can undergo torpor. And so you see the title is Daily Torpor in Mammals. This is essentially all of the branches of the mammalian tree, or a huge number of the branches of the mammalian tree, going all the way back to monotremes and marsupials. And so these animals actually existed before placentas evolved, and they already knew how to undergo torpor. So this torpor is very ancient. So a quick quiz. We're looking for a mammal here. It lives in Tasmania. It eats ants. It lays eggs, and it hibernates. If you guessed Tachyglossus aculeatus, you are a nerd. But you're correct. And if you guessed the echidna or the spiny anteater, you also get all of the points. And look how cute that thing is. You can see, even though that animal lays eggs, you can see in its eyes and its cute little feet, like we recognize that kind of as us, right? Like that is much cuter and cuddly than a baby chicken or a baby lizard, even though baby chickens and lizards are cute too, right? But there is something mammalian in that we recognize as being part of our tribe, I would argue. Look at that little tongue. So mammals who undergo torpor predate placentas, as I've said, and I think that's very interesting. That suggests that before the first mammal evolved, something like the echidna that produces milk, and that has fur, but has not yet evolved a placenta, already had the genetic program to undergo torpor. So it had already evolved a hot-blooded metabolism, but it also maintained a switch to turn it off in times of scarcity. Also looking at this mammalian family tree, mammals who undergo torpor includes primates. So torpor is conserved all the way to our branch of mammalia, which means mammals evolved about 180 million years ago. The first mammals had torpor. They lived throughout the entire age of the dinosaurs. After the dinosaurs went extinct, there was this huge time of evolution of mammals when all these new kinds of mammals evolved. Torpor survived that entire time, and it made it until the first primates evolved. And so we split off about 55 million years ago from lemurs, and we're going to talk about lemurs in this episode. And so, yes, torpor survived through 120 million years throughout every branch of mammality, if I can use the word mammality, and even onto the branch of primates. So it seems to be a pretty well-conserved thing that mammals have the ability to do. And it's not just me saying this in this series. I like to follow the money. So the space agencies, you can see this work was supported by the European space agencies. So the space agencies of the world are spending money to try to prove that humans can indeed hibernate, and that humans have this whole program of torpor. So torpor is an alternate metabolic state where you can slow down your metabolism, lower your body temperature, and also where you can preferentially store fat rather than burning it. And torpor is necessary to hibernate. Torpor and hibernation are not quite the same, but you have to undergo torpor to then undergo hibernation. And the space agencies of the world are spending money with the idea that humans also have this mammalian program to hibernate. So I think, it's a good bet that humans can and do undergo some type of torpor. And so these are the primates who undergo torpor. This is the dwarf lemur of Madagascar. It's a very interesting animal because, again, it's the closest mammal to us that is known to hibernate. And it lives in the tropics. And so oftentimes we talk about hibernation and torpor as being something that happens in the far north and winter's coming. But in fact, it's torpor is more clever than that. It's using all of these signals from the wild to know that winter is coming, because presumably these small animals like squirrels are not waking up thinking like, oh no, winter's coming, I have to do all these things. The squirrels are just doing what feels natural to them in that day, right? They're following their instinct. And they are, in the course of that, they're fattening up for winter. What they did in this study is they looked at the microbiomes of these tropical hibernating primates in the wild. And so the reason that they're hibernating is that the dry season is coming in this case. And so lemurs hibernate through the dry season. And this study looked at the microbiomes of these lemurs during the fattening season. And this is important. And a lot of studies about torpor look at the animals sort of like while they're in torpor, and not necessarily, they sometimes miss that crucial season in the north. It would be like in October as they're fattening for winter. A lot of the times the papers miss that season, which is unfortunate, because that is the season where they kind of resemble obese humans, and is, I think, a really useful analogy for us. And this is what they found. So these are the lemurs in the fattening stage. And so these are all different clans of bacteria. They're not species of bacteria. These are like families of bacteria. So when you look at this bacteroidetes, there's probably thousands of bacteroidetes species, or millions or billions, I don't know. And you can see that in the fattening stage, you don't see a lot of blue. So each of these columns is an individual animal. And like I say, these are wild collectors. So they had to go out into the wild and trap these animals and get fecal samples and test their microbiome. So this isn't that easy to do. And so these columns are all individuals. And some of, they didn't get all the same animals in the fattening time. And then emergence, this is like summer, this is like spring, this is when the animals are done hibernating, they're coming out, they're in the lean part of the season. This is probably mating season. They're probably listening to music, dancing, going to festivals, all of the things that we do in the most active part of our year, right? And so you can see that you can see a couple things. One, in the emergence season, you see a lot more light blue. And in the fattening season, you see a lot more green. And so in some of the individuals that they got in both seasons are the same. So like, for instance, the one I underlined in green, FA, this is one animal in the fattening season. You can see that it has, it's got this much green. And this over here is the same animal in the summer. And you see the green goes way down. And this bacteroidetes is not evident during the fattening season, but in the summer, look at that. It's got the bacteroidetes have returned. This RA animal is very interesting. So during the fattening season, it has tons and tons and tons of firmicutes, and a very small amount of this bacteroidetes in the summer, in the, dancing and festival part of the year. The firmicutes is, very small, and the bacteroidetes are very plentiful indeed. And so you see the shift when they're in the fattening part of the season, you see a lot of firmicutes, and when they're in the lean part of the season, you see a lot of these bacteroidetes. And so I made a little mnemonic. So remember this: fattening is firmicutes, and when they're bean poles, when they're lean in the summer, that's bacteroidetes. I know it's a very clever acronym. Anywho, and so is this relevant to humans? So this is a study out of Japan looking at obese and lean humans, and you can see that the bacteroidetes are about 35% in the lean humans, and they're all the way down to 23% in the obese humans. And so you see that pattern of less bacteroidetes, less of the beanpole bacteria in the obese humans, and that makes sense. and this is in the Ukraine, and the same thing happened. So these lean people are actually quite lean. They have a BMI of under 18.5, so they are kind of literally beanpoles. And you can see they have a lot of bacteroidetes. These obese Ukrainians have a BMI of over 30, and you can see they have significantly less bacteroidetes. They also have a lot more firmicutes, the bacteria that you see in the fattening season. So obese humans and fattening hibernators have a low ratio of bacteroidetes, the beanpole bacteria, to firmicutes, the fattening bacteria. And I do want to say this has not consistently been shown in human studies. Humans are a very heterogeneous bunch. We do all kinds of dumb things and crazy things. And you know what all those dumb and crazy things are. Swigging down soybean oil, staying up late, partying and drinking, or just, sitting around all day fretting about our ex or whatever's bothering us. And all these things have effects, right? And so this hasn't been consistent in humans. And I also want to say that I don't think that the bacteroidetes, firmicutes, like I say, there's like thousands of species of each of them. I don't think this is a hard and fast rule. I think it's an interesting indicator of what's going on, though. And so one of the questions is, does our microbiome cause our obesity, or is it a symptom of our obesity, right? And so this is a very interesting study. It was in the journal Science. What they did was they found identical twins, and so they have the exact same genetics. They, developed in the same womb, so they presumably have the same gut microbiome at birth. They have the same epigenetics, and then later in life, one twin became obese and the other one did not. And so what they did was they took these germ-free mice that don't have a microbiome, and they put bacteria from the obese twin in one, and they put bacteria from the lean twin in the other one. And you can see that the mice who got the bacteria from the fat twin indeed increased their fat mass accordingly. So this suggests that the microbiome of the fat twin is indeed causing, weight gain, or at least some of the weight gain. And the bacteria from the lean twin does not cause weight gain. And so then, that's very interesting, right? What affects our gut microbiome? Things like fiber. Perhaps the reason that both the lemurs and the obese humans, maybe the lemurs, something's changed in the environment and there's not enough fiber, and that's why their microbiome is different, and that's helping them to fatten, right? Same thing in the obese people. Maybe they're not eating as much fiber, just as a for instance, right? Theoretical. We also know antibiotics affect the gut microbiome. You can just, essentially napalm your entire microbiome by taking enough antibiotics. And then there's things like prebiotics. Prebiotics are a bit like fiber. There's specific things that you can eat that are known to sort of feed certain bacteria and to promote certain healthful bacteria. And probiotics just means eating actual bacteria, things like yogurt, things like sauerkraut, and hoping that there are bacteria in there who survive the stomach acid and go on and colonize the intestine with more beneficial bugs, right? And this is a study out of Korea, and so they took this NR28 is a strain of bacteria that came out of kimchi, actually. And you can see that when they gave this kimchi bacteria to these mice, the firmicutes went down, so the fattening bacteria went down, the beanpole bacteria went up, the bacteroidetes, and they have less fat. And so this is a nice example of a probiotic shifting the gut microbiome in a favorable fashion and the mice losing weight. And another thing that affects the gut microbiome is the idea of fecal transfer. And so the former study, so this is the same study that I showed you before with the identical twins. And what they also did was this is a fun little experiment. So these first two columns are the two columns that I already showed you. But what they also did was, so they took mice that got the bacteria from the fat twin and mice that got the bacteria from the lean twin, and they put them in the same cage. And so these two blue columns, where you see some fat gain, they both started out with the bacteria from the fat twin, and they both gained a little bit of weight. The blue column, it's housed alone, and this column where they gained significantly less weight, they were housed with another mouse that got the microbiome from the lean twin, right? And one of the things about mice is they practice something called coprophagy, which means eating poop. So if you put a fat mouse and a lean mouse together in a cage, they will eat each other's poop. And something about being housed with the lean mice caused the fat mice to not gain so much weight. And what they concluded is that the transformation correlated with invasion of members of bacteroidetes, which of course are members of the bacteroidetes clan, from the lean mice into the obese microbiota. And so what you see is that the beanpole bacteria have migrated from the lean mice to the obese mice and helped them not gain so much weight. Like I've said, fattening is the biological imperative of the hibernator. Specific bacterial strains cause fat loss, as you saw in that last experiment. And so I ask you a question. If you were the thing in charge of making sure that the organism fattened for winter, would you leave the microbiome diversity to chance? And like I say in the last episode, obviously I argued that the Aryl Hydrocarbon Receptor is the, or at least a, master controller of torpor and fattening. And so this paper is very good about bidirectional communication between the Aryl Hydrocarbon Receptor and the gut microbiome and the host metabolism. And what they showed is that, so this is in mice, and this is the gut microbiome of mice on a normal diet. And in fact, this diet was specifically selective to not have anything in it that was a known activator of the Aryl Hydrocarbon Receptor. And in this diet, they actually added an Aryl Hydrocarbon Receptor activator to the diet. And you can see what happened. The firmicutes, the bacteria involved in fattening, absolutely explode when the Aryl Hydrocarbon Receptor is activated. And the bacteroidetes, the beanpole bacteria that you see in the summer, in the spring, in the lean periods, is almost completely eliminated if you activate the Aryl Hydrocarbon Receptor. Just to show that's not a one-off. This is a second study. It's again done in mice, but these mice are kept in a cage, and you see they gave 4 different groups of mice increasing concentrations of TCDD, which is another thing that activates the Aryl Hydrocarbon Receptor. And they did this over 28 days, over about a month. Every 4 days, they just injected into their mouth, essentially, with like a little bit drop of corn oil, this TCDD. And you can see that, so this is a dose response. So the more TCDD they got, the more their Aryl Hydrocarbon Receptor was activated, the less of the bacteroidetes, the beanpole bacteria there were, and the more of the fattening bacteria there were. And so this shows that the Aryl Hydrocarbon Receptor is somehow, and we don't really know how, is actively controlling our gut microbiome to favor the fattening process. And that is very interesting. And this is a slide from episode 4, but these obese humans look like fattening hibernators. And the point here is this kynurenine is it's a tryptophan metabolite, but it's something in our bloodstream that activates the Aryl Hydrocarbon Receptor in the liver and in other tissues. And so you see that in obese humans, you have about 50% elevated kynurenine, and that means an activated Aryl Hydrocarbon Receptor. And you see in hibernators, in summer, the kynurenine is at its lowest, and in the fall, when they're fattening, kynurenine is at its highest. And so the fattening hibernators also have an active activated aryl hydrocarbon receptor in the fall when we know that they have a lot of fattening bacteria and not many beanpole bacteria, right? And so this gives me the idea of active versus passive control of the microbiome, because there are ways we can actively control our microbiome, like antibiotics. Obviously, we can just burn down all of our microbiome. That's a very active strategy towards controlling our microbiome, right? But also, I think the aryl hydrocarbon receptor belongs in this category, because the aryl hydrocarbon receptor is really it is going out and it is changing the gut microbiome very actively, as opposed to things like probiotics, prebiotics, fiber. These are strategies where, the idea is, okay, if we feed our microbiome and give our microbiome the things that it needs, it will respond in a favorable direction, but it's kind of passive. We're sort of feeding it and hoping it for the best. And I put fecal transfer there. Fecal transfer probably falls, you could put it in either category. I'm not sure. But what I would say is, if I was going to do a fecal transfer, I would hope that first I had my aryl hydrocarbon receptor under control, because otherwise it's just going to eliminate the good bugs anyway, right? And so I just want to show that indeed, so the Western diet activates the aryl hydrocarbon receptor, right? And so this was shown by the Tomlinson group. They fed these mice a Western diet. And this gene CYP1B1 is actually a target gene of the aryl hydrocarbon receptor. And so the AHR is tricky to measure, because it sort of doesn't matter how much of the protein there is or how much of the mRNA there is. What matters is whether or not it's activated. And so the AHR turns on CYP1B1. And so that's the best way to look whether it's activated. You look at the genes that it controls, and if they're turned on, then it's probably activated. And so when we fed these mice a Western diet, you can see CYP1B1 is increased by almost 2.5-fold. And we know that this is being done by the aryl hydrocarbon receptor, and that CYP1B1 isn't being turned on by some other thing, because if we give these same mice on a Western diet an aryl hydrocarbon receptor inhibitor, CYP1B1 is not increased. So AHR is increased by a Western diet. And so here I'm going to talk again about poor T. So you'll see a theme in these videos. The AHR, the aryl hydrocarbon receptor, is doing something bad, and somehow poor T is preventing the bad thing, right? And so I want to talk about how poor T is reversing the effects of this AHR activation by the Western diet, right? And so here you see these mice, they're on a Western diet, they become very fat. And this is those same mice given the Western diet plus the poor T, and you see that they don't become nearly as fat as the mice on the Western diet. And what's interesting about this study is, here's what happens to the gut microbiome, right? The mice given the Western diet have this huge increase in firmicutes, the fattening bacteria, which is exactly what you'd expect if you have an activated aryl hydrocarbon receptor, right? And the bacteroidetes are very much minimized, which is, again, exactly what you expect if you have an activated aryl hydrocarbon receptor. The ones given the Western diet plus the poor T, you can see do not have nearly as big an increase in firmicutes, and they manage to preserve more of their bacteroidetes. And so the something about the poor T is preventing the worst effects of AHR activation by a Western diet on our gut microbiome. And so this is in the same theme, but you saw in episode 2, the AHR decreases bile acid excretion. Theobromine from poor T increases it. AHR activation dysregulates the circadian rhythm. The poor T restores circadian rhythm. The AHR decreases, in this episode, the bacteroidetes to firmicutes ratio. And in this episode, the poor T increases the bacteroidetes to firmicutes ratio. This is why I sell a theobromine extract of poor T on the blog at fireinabottle.net/shop. I only have a few products there. I try to find products that are really unique and that I absolutely believe in before I'll have that product. And, if you get that product, obviously it helps me make these videos that I hope are informative. I hope also that Tia Browning extract is really good for you and helps you out, because you can see, as I think, it's doing a lot of good things. Also, if you want to help the blog, this t-shirt, this is a deeply philosophical t-shirt, wrapped in a dumb joke. So this is a dwarf lemur. This is the closest mammal related to us that hibernates. This is the echidna, and of course it's more high res on the actual t-shirt than it is on this screenshot I took. This is the echidna, the furthest mammal away from us, and the phrase "you a mammal, bro" is questioning that, we are all mammals. We seem to all have this genetic heritage of the ability to undergo torpor, that the space agencies of the world are spending real money on trying to figure out how to unlock that program. And you probably do not want to be in torpor as an active human with a day job, I would argue. And so we're going to, in this video series, we're going to continue to think about how we get out of torpor and what are the signals that bring it on and what are the signals that turn it off, other than just puberty, although the puberty is important. And so conclusions from this video: fattening is the biological imperative of the hibernator. Otis needs to get very fat to survive winter, and so it is going to do everything it can to make sure that it is storing calories and not burning them, right? The AHR manipulates your microbiome to favor fattening. And again, if that AHR is activated, you've got all of the firmicutes that you see in the fattening mammals, and you've got none of the bacteroidetes that you see in lean mammals, because the AHR, its job is to get you through winter and make sure that you fatten up, right? That's its goal. And so it manipulates your microbiome for that purpose. Lastly, probiotics are great, but you want to make sure that you have the AHR under control, I think, before you start thinking about probiotics and prebiotics. And so I would argue that, maybe start with the puberty, try to get the AHR under control. I'm going to have more ideas about getting the AHR under control in the next few weeks. And then hit it with the yogurt and the sauerkraut and the kimchi. Next episode, come back soon. I've got a whole bunch of episodes planned for this winter and this year. We're going to talk about using oxidants to fight oxidative stress. Yes, you read it right. It's not a typo. We are going to talk about how oxidants are the things that prevent and restore oxidative stress to a proper redox value, because you're all overly reduced and you need to get out of that reductive stress using oxidants to get rid of your oxidative stress. As counterintuitive as that may seem, that is the truth. In the meantime, check out fireinabottle.net/shop, get some Tia Browning extract, and I will see you next time.